All right, promise. Last podcast for biotech should be pretty short. We have talked about all the different ways you can cut up and analyze DNA. Well, not certainly not all of them. Some of the most important ones for you to know for our purposes in this class. You can cut up DNA and actually sequence the order of the A's, T's, C's, and G's. You can cut up DNA to find a specific gene and in order to reproduce it or put it in a different organism. There's all kinds of reasons and ways to cut up DNA and things you can do with it. What we are going to talk about now is how you can cut a gene out of one organism and insert it into another and make that organism do something that it never could do before, okay, and likely would not do. You couldn't crossbreed to get this result. It certainly wouldn't likely happen naturally unless you had a really bizarre mutation all of a sudden, in which case maybe you could get glow in the dark, whatever. No, I don't. Okay, what we're going to talk about is basic genetic engineering, and this is part of the lab that we are going to do. We are going to genetically alter bacteria, and this is going to kind of get us into the basics of how we're going to do that. Okay, so if you'll recall, we talked about plasmids being used as vectors to take DNA from one thing and put it into a bacterium, for example. Okay, so we're going to talk about that in a little bit more depth. Let me get to these. Okay, restriction enzymes. We've talked about restriction enzymes. I do want to remind you about the sticky ends that restriction enzymes can have. That's really the only kind that's useful to us in biotech, at least certainly in genetic engineering, are the sticky ends, where if we cut here, we're leaving open nucleotides there and open nucleotides here that will readily bond with complementary nucleotides. Okay, so what we would do in a case of genetic engineering then is we would find a gene that we wanted, perhaps for human insulin, like we talked about, or perhaps for glowing in the dark. And we would cut it out of the organism in which it existed. We would cut the piece of DNA right out of there, and it would hopefully have those sticky ends. And we would cut another piece of DNA with the same restriction enzymes, so they have the same kinds of sticky ends. They have complementary sticky ends, not only on the little fragment of DNA that has the gene we're interested in, maybe it's the glow in the dark gene, but in the larger piece of DNA, maybe right here, we cut it right there with the same restriction enzymes, so that now this thing is open and it's got sticky end that matches this sticky end and that sticky end, so that if we just could get the two together, they'd stick right together. Okay? So there's the gene you want with its sticky ends. Here's the chromosome you want to add the gene into. So this would correspond with that, and this would correspond with that. Okay? And when we combine them, we get that connection. That would represent 1/2, like, whoops, probably this fitting right in there. Okay? And then we aren't seeing this end that's attaching to that. Okay? So sticky ends help glue genes together. Okay? We cut out the part we want. Here's the gene we want right there. We cut it out. There it is with its sticky end. Here's the chromosome we're going to add the gene to. We're going to cut that and stick that gene we want right in there. Okay? Please remember, this guy, DNA ligase, is used by scientists, biotechnologists, to join the strands together once they are added to the same pot. This end result is called a recombinant DNA molecule. It is a new combination of DNA that did not exist before. you are made of recombinant DNA. You're a new combination of your mother and father's DNA that did not exist before you did. Okay? So, when these two sets of genes, the large genome and this particular gene of interest that we added to it, are put into an organism, then this gene should produce the protein that it codes for. So, for example, if this were the gene for human insulin and we were sticking this gene into the chromosome of a bacterium, then that bacterium would produce human insulin. Pretty nifty trick. Okay, all of a sudden we can get human insulin produced in vast quantities by big vats of bacteria in some pharmaceutical company somewhere. Okay, so, and there you go, from bacteria to little bottle to customer. Okay, now, this is a really good question. How in the world can bacteria read human DNA and make the same protein that a human would make? Well, they can, because remember, this code is universal. All living things use the same code. Incredibly important, not just because that means we can use it in the way we just illustrated, but because it shows that all life does have a very deep rooted connection in this genetic language that we use. Okay? All right. Now, what we will do, okay, so we created this new piece of DNA, this recombinant piece of DNA, but we haven't addressed getting it into the bacteria. That requires this idea of transformation that we talked about earlier. This is where we want to insert that new DNA combination into a bacteria and hope that the bacteria will work. We're going to grow that bacteria in cultures, okay? And that bacteria, when it is growing, is going to make lots of copies of that gene. And so essentially it's going to be cloning that genetically engineered plasmid that we made and make tons of copies of it. Plus, while it's doing that, it's also going to be making bunches and bunches of the new protein that we want. And we are going to have a completely transformed bacteria. It's going to have a new physical characteristic. It's going to have a new phenotype, if you remember your genetic language. Okay? I think this is just an overview of what we just talked about. So here is your bacterial plasmid. Remember, these are always our vectors. We use our restriction enzymes to cut it open. We cut the gene from the other organism, perhaps human insulin, perhaps glow-in-the-dark from a jellyfish. We stick this gene with sticky ends into our plasmid here, and so what we end up with now is a vector that's got the new gene in it. Okay, a recombinant plasmid, a new combination. We stick this or encourage the bacteria to pick up these plasmids, and this is what we are going to do in class, and then we just grow piles and piles of the bacteria. And then what we need to do, if we want to collect the protein, if this were human insulin, and we wanted to collect it, we would have to figure out a way to harvest and purify all the protein, the insulin, that these bacteria are now making, and certainly they do that. Uses of genetic engineering that we touched on at the beginning of all this: genetically modified organisms, GMOs. There's also GMF, genetically modified food, which is just kind of an offshoot of genetically modified organisms. We can engineer plants, corn, rice, whatever, to produce new proteins that they didn't produce before. Sometimes these protect crops from insects. The transformed corn can produce a bacterial toxin that is going to kill this guy that wants to eat the corn in the field. So it kind of is making its own pesticide, if you will, that it evidently is not dangerous for anything but this unsuspecting caterpillar. We can engineer plants to extend the growing season. Basically, for example, strawberries that have an kind of an antifreeze gene from flounder can be grown when it is generally too cold to grow normal strawberries. Improving food quality. This is of huge importance in places that are struggling to have enough food to feed the people that live there, developing countries. This idea of this golden rice, it has got a higher nutrient concentration. Producing vitamins that are not found in large amounts in normal rice, and thus making it higher nutritional value. Okay, this is what we're going to be dealing with, however. These jellyfish have a gene called GFP, green, I believe that's green fluorescent protein. We can take that gene, and not only can we put it in bacteria, but we can also put it in mammals. We've put it in mice and made them glow in the dark, and Mr. Pig and all that. And these are called genetically transformed organisms. They are transformed. Pretty amazing stuff. So that'll do it. I told you this one would be shorter. I thank you for sticking with me, and I hope we will. This now means we'll be doing our labs. So thanks for watching. I'll catch you later.